Hello to everyone. Welcome to my talk about CRISPR-Cas, these gene scissors. And I want to introduce myself. My name is Maricel Santoro. I come from Argentina. I've been living here in Germany for a few years. And now I'm doing a postdoc in the Natural Product Biosynthesis department in the Max Planck for Chemical Ecology. So let's start with this Let's CRISPR, an introduction to the gene editing methodology. So why this manipulating DNA is so important, why the DNA is so important? And since we know a lot of organisms in the world, like most of it, like plants, or animals, and bacteria, and fungi, they all have DNA. And the DNA is actually stored in the cells and in a small compartment, which is the nucleus, and is coiled, and it's coiled to be able to fit in this compartment. But if you uncoil it, we can actually see the sequence of this DNA. What is this sequence? It's just a thread. If you want to think about it, they are thread with letters. These letters are very important, and it's like 4 possible letters that we can have in our DNA, which is thymine, adenine, guanine, and cytosine. They come in couples, that's why they are called base pair. So adenine is always like related with thymine on the other strand, so it's 2 thread, and guanine with cytosine. And this it's important because this is information which is actually then generated from the DNA, generating RNA, and this information is like transported to the cytosol, where this DNA is actually transformed into proteins. So these proteins are very important because they are like they can synthesize enzyme or the structure of the cell, and these enzymes is like they are the workers of the cells. They are just produce all the compounds that are present in the cells. They are synthesized by enzymes. And if you can think about it in a more simple way, we can think about the DNA as a list of instructions that we have in our cells. There is actually, via a messenger with a process that is called transcription, it's actually transcribed in RNA, and this RNA is translated afterwards to protein. This translation is based on the genetic code. And this sequence of these 4 letters can be translated into these units of the proteins, which are the amino acids. These proteins are then condensed into enzymes or structural proteins. And we can think about these enzymes as the workers of the cells. They do everything inside the cell. They synthesize all the compounds that are present in the cells. So what do you think about genetic manipulation? What do we think about it? And if I was searching actually online about what people think about genetic manipulation, and actually not good things came out. And people are scared about it because they don't know what is happening. And the idea is that we can actually generate strange living forms. Like, for example, an orange with a kiwi, a form of an orange with a kiwi inside, like animals with different characteristics. For example, if you are like fan of the comics, like Hulk is also like generated by genetic manipulation. But actually, this can be used also for good reasons. We have been manipulating the DNA of organisms, and mainly in plants, because that's what we work here in Max Planck. We work with plants. And we have been doing this for centuries. And why? Because we want to improve, for example, characteristics of certain organisms. For example, the mice that we eat today, it wasn't like that like several years ago. So if you see that, actually the fruit of the mice was very small, and it was not flashy at all. So through breeding, so crossing and then selecting a better plant, and then crossing, and through centuries, we had this kind of very good and tasty mice that we have today. And the same thing happens, for example, with banana, which is an hybrid, seedless, and before also the white banana actually is like that nowadays too, with a lot of seeds and probably not tasty, not with a lot of sugars. The same happened with the watermelon, with the peach, with the eggplants that didn't look at all at the eggplant that we have today. So, and for example, carrots also, they didn't look as the tasty carrot that we have today. So everything has been done to improve qualities, to reduce, for example, bitterness in the fruits, to improve, for example, the water content, the fleshy content, so this meat that we have, the flesh that we have in the fruits. So this has been done without knowing what we were doing. Centuries ago, we didn't know what we were doing, and we were actually selecting better DNA in the plants. So with time, we actually realized how this was working, that the DNA actually stored all the information. So we start actually manipulating this DNA. And the first technique that we had is the mutation breeding. So we were using agents to mutate the seeds or to irradiate the seeds, for example, X-rays or gamma rays, like the Hulk rays, or chemical compounds like EMS. So we were submitting these seeds to these agents and then making them grow and then crossing them. And with several generations afterwards, we could select the plant with an improving trait. So, and this has been done, and we actually have these crops nowadays. For example, rice has been done like that. There is several actually cultivars that are around and used nowadays because this is actually permitted. Oranges, peas, and for example, tomatoes. And all of them have been doing for improving resistance, for example, to pathogens, to herbivores, or to improving the qualities of the fruit. So, but for example, this wasn't enough because we have also realized that the frequency of this mutation is very low and the screening is very laborious. You have to wait several generations afterwards to have an improved plant. So what we were doing, for example, another technique was a DNA recombinant technology. So some organisms have some characteristics that we like, and for example, we want to put in another organism. For example, bacteria has a lot of resistance to herbicides and to a lot of antibiotic resistance. So this is a very good quality that we could improve our, for example, our crops. So what we do is like take one organism with the gene of interest and then put it inside another organism. This is one example how we do it with plants. So we have one bacteria which is normally infecting plants, generating small tumor. Probably you have seen this, Agrobacterium tumefaciens, results in plant. So they carry a vector, which is also a DNA molecule, in what we want to put our gene of interest. And then this vector with this bacteria is going into the plant. And then we have in the plant the gene of interest that we want. So this was actually achieved after the discovery of very important enzymes, which are the restriction enzymes. The restriction enzymes are enzymes that cut specific sites in the DNA. Every restriction enzyme recognizes a site and then cut it. This cut can be sticky, so one strand of the DNA can be like protrudent and the other one not. So we can actually put these two together if the sequence is actually homologous, and then we can have a recombinant DNA. This is one technique that we use to clone different genes inside other organisms. And actually this has been used in crops, and one example is the maize, the MON 810, or the BT maize. Some people know it like that. There is a resistant to biotic stresses, so there are a lot of pests. They can have deleterious effects on the crops. So we actually, what scientists did, so they took this protein produced by a bacteria, Bacillus thuringiensis, and they put it inside the maize. So the maize actually is resistant to these biotic stresses, so we don't lose crops. But another example, for example, is on potatoes, which have been, the starch in potatoes is a very important industrial characteristic of the potatoes. And there is, the starch is made by two components. And the one with the industrial actually interest is the amylopectin. So they produce potatoes with more amylopectin. And this was actually used for several years, but now it's banned. It's not used anymore because the selection of the plants which actually carry this external exogenous gene, they have to have a marker, and the marker generally is antibiotic resistant, and this is actually coming from a bacteria, so it's not well accepted for the community. Another example that everyone knows is the golden rice. The golden rice is just rice that produce another component, which is beta-carotene. Beta-carotene is a provitamin A, can be used to produce vitamin A. So what people did, they took this beta-carotene, 3 genes from the tomato, to produce beta-carotene in seeds of the rice. So it's actually a good source of provitamin A. This is also highly regulated, and not so many countries allow the use of these crops. So the problem with the DNA recombinant technology is the regulation, and we don't know why there is so many regulation about it, because we don't know the interaction of this foreign DNA that we put in the plant with other organisms where the plant actually lives. The plant interacts with a lot of organisms, for example, bacteria in the soil, worms, insects, and pollinator insects. So we don't know this interaction, the effect that we can have in the ecosystem. So this is not very well known, very well seen actually. And but we still keep wanting to manipulate DNA. We'll keep finding new technologies to manipulate DNA. And why this is so important, because there is one mechanism to understand the functions in a cell, which is based on the gene sequence. So the tool that we have as living organisms is our DNA. Our DNA codify all the functions that we have. So it's very important to know the functions of the genes to be able to cure diseases, to be able to improve characteristics, to be able to make our life better. So the known methodology to actually understand this is the forward genetic, is the old genetic. So we know a phenotype. For example, one plant is more resistant to an herbivore. So, and we want to different methodologies understand which is the gene responsible, one or several genes responsible for this feature. But nowadays, since we have a lot of DNA sequencing, a lot of organisms, actually we know the sequence of the DNA. There is this the other side of the coin, which is the reverse genetics. So we know the gene, and we want to know which effect this gene has in the plant. So one of the tools that we have nowadays is like recognize which gene could be responsible for a trait. So this is known for the relation of the plants between them, the study that have been done before. So we can target that gene and see, for example, if we delete the function of the gene in a plant, see what's happening in the plant. What is less present in the plant? Is the plant more sensitive or resistant to some other effects? So these two methodologies have limitations. For example, this mutation breeding has a limitation of being random. So we irradiate a plant with X-rays or gamma rays, or we just put the seeds in contact with some chemicals, mutagens chemicals, and the effects can be random. We don't know where this mutation is actually occurring. And the DNA recombinant technology, also, there is a random insertion where we put exogenous DNA in a plant. We don't know where it's going to actually insert in the DNA of the plant. And there are limited applications because we cannot use it to put, for example, we can just insert a gene in a plant, but we cannot modify all the genes that are present in the plant. So we needed actually a tool that was more specific for the sequence that we want to actually act on it. And so that's why the gene edit came. The gene editing is a very useful and actually very flexible tool, and it's a precise modification in a gene sequence. So we know our sequence, and we want to modify to see what's the effect that we have in the plant afterwards, or in the organism afterwards, to know the function. So what the gene editing has two actually components: an enzyme, so this worker, and which is called Cas9, and an RNA. there are two different kind of molecules. The RNA is very similar to the DNA, and the enzymes are this protein that actually made all the reactions in the cell. And the RNA is called guide RNA, and it has two components too. There is a target sequence, which is actually a specific sequence that we can modify, depending on which gene on the organism we want to target or mutate. And there is another part of it which is called scaffold, which interacts with the enzymes to actually guide this worker to do to actually act on the DNA. So we can think about the guide RNA as a GPS, which is actually guiding this worker with scissors that is gonna cut our DNA or mutate our DNA. So these two components interact, as I told you. The scaffold interacts with the Cas9, and then the target is actually available to find the place in the genome, in the DNA, to find the gene in the DNA that it wants to mutate. So that is our target. So once it finds the target, so the target of the guide RNA, this target sequence actually assembled via homology to the DNA of that we want to work, and in a specific place, which is actually the site is actually is given by the target of the guide RNA, and also another specific sequence, which is the PAM sequence. It is the only sequence recognized by the enzyme. So once they are in the correct place, there is the cut. So these scissors cut both strands of the DNA, generating a double-strand break. This double-strand break is toxic for the cells because there is an interruption in our DNA sequence, so the cells has to repair it. And it's repaired by two main mechanisms, which is a mechanism which is random repairing. So there is this double-strand break, and I have to bind it again. So this binding or this ligation is actually done with a loss of some of the nucleotides that are in the DNA, or the addition of some nucleotides. This addition, these indels, they are called, so addition or loss of nucleotides, it generates a mutation in the DNA, in the gene sequence. Another mechanism is based on homology. So if we give actually to the plant a donor which has a similar sequence to the overhang on the double-strand break, we can actually insert DNA in this place of cutting. So we can actually mutate a gene to annul the function, or we can actually insert a new gene in the DNA. So this is very important, and it's because the interaction between the guide, so this GPS, and the protein, so this worker, is based on an RNA-DNA interaction. So the DNA and the RNA are very similar molecules. They are nucleic acid molecules, so they can interact very easily, and it's very easy to predict where, the interaction. So this double-strand break, the generation of this cutting, is not actually a new idea. So it's not something that we discover with this gene editing, actually with the CRISPR-Cas gene editing, but it's something that was already going around in the groups that were doing actually manipulation of genome, and they were using different mechanisms. Meganucleases, they are like restriction enzymes, like, but they recognize very big actually sites to cut. They're usually present one time in the genome, so, and they has to be present in the gene that you want to modify, you know. So it's not there is some limitation of the applications that we have. So afterwards came the TALEN and the zinc finger methodology. They actually generate a target with a protein part, which is the TALEN or the zinc finger, which is this one or this one. But this interaction is mediated, is actually protein DNA interaction. It's very hard to predict, and it's very hard to build. It's very expensive, and so it wasn't like the applications were very low, and the efficiency was very low. And also these meganucleases weren't like a very wide use, because the recognition site is in the enzyme, so we cannot change it. So then this CRISPR-Cas system came, but it's not that the scientists were in the lab and just decide to invent this system. Actually, they discovered the system in bacteria. Bacteria also have a defense, like we have, against diseases. So they have an immune system. They defend themselves against viruses. And they come, for example, in contact with the virus first time, and then they have a memory about this DNA that was present in the virus. And then the second time that they encounter the virus, they can defend themselves. So this system has spacers and tracer DNA. These two components are what nowadays we call target, so which is specific for the sequence that we want to modify, and the scaffold, which was interacting with the enzyme, which are the Cas protein. They were also there in the bacterial genome. So then the modification of this system and why it actually earned a Nobel Prize this last year was because this was, they make it simple. They make it simpler because these two pieces of RNA, they make it in one piece of RNA, adding a loop here. So it's easy to build, it's easy to produce, and it's low costing. And studying this system plus this application, so these two scientists like Emmanuel Charpentier and Jennifer Doudna just earned the Nobel Prize last year for this methodology. And this was discovered in 2012. And since 2012, the application has been you cannot count them. They are like every day coming out papers of application of CRISPR, not just, for example, in plants, in animals, in bacteria, in fungi. So it's a very wide use, and it's very easy to build, and it's very cheap actually to build. So in plant also there have been some application of it. For example, in Germans will like this. So in cucumber, there was some application of the numbers of actually fruit was increasing during a CRISPR knockout. So there is a gene, this VIP gene was actually inhibiting, or like I would say, limiting the number of flowers that there were in the plants. So actually deleting the function of this gene make the plants producing more fruits. Another, for example, the ground cherry, that is also very well known here in Germany. So there was another knockout of the plant. The plant was actually very high, growing more in height and producing just 1 or 2 fruit per plant. So they actually knock out another gene with the self-pruning gene, which increased the number of fruit per nodes. So increase the number of fruits, so we have more yield. In tomato, for example, there's a problem with tomato. I don't know if you cultivate tomatoes in your gardens, but they are very fast softening. So if you keep them at home, they can actually go bad very quickly. So if you, for example, knock out this gene, so they can actually last a bit longer, like we can see here in the image. There is the wild type and there is the knockout plants. Another example, for example, is kiwi. So the kiwi is it's a plant also that grows high, more than generating more fruits. So there was another knockout in this gene. So generate compact plants. This is a normal kiwi plant, and these are the compact plants. So if you can see, they have more kiwis, per node, and they can actually rapid flowering and generating the fruits faster than the wild type. So this is the gene editing, when we actually edit a gene, so a sequence which is codifying for a protein. So what is happening when we want to modify other sequences? So in our DNA, in our very long sequence of DNA, not just ours, but every organism, there are other sequences together. It's not that we have the gene and that's it. We have to regulate the expression of the genes, for example. So they cannot be all the genes active at all times. So we have other sequences close by. So we have, for example, a promoter, which is the regulation of the genes. So this is like regulating if it's active, the transcription or the production of this gene, or is not active. And this is actually the codifying sequence of the gene, which in higher organisms like animals and plants, we have this kind of interruption in the sequence. So we have spacers and we have codifying sequences, which are called exons, the codifying sequence, intron, the spacers. They can be very useful. It's not just because they are there, but sometimes one gene can be, then these spacers, for example, are taken out, and we have the complete codifying sequence. Sometimes this splicing, which is this improving the sequence, taking out these spacers, can generate from one gene different CDS, so different genes. For example, not putting this exon, or for example, avoiding the last exon. So we have from one gene, we can have different products. So this is also one characteristic. And so we can think about it, as I told you before, the DNA or the gene is our instructions, and we can think about the promoter as a switch. See if it's switching on or switching off. There is another mechanism of regulation, which is we can actually regulate in the sequence when we decorate the sequence with the methyl with some other chemical groups. For example, we methylate the sequence. Is another regulation. So since we have other sequences that actually participate in the function of the genes, like for example the promoters, so we cannot cutting a promoter or cutting a sequence is the same, but sometimes we don't want to get rid of the sequence of the gene, but we want to know what the gene is doing in certain conditions, so we can decide when to turn on a gene or turn off a gene. So if we want to do that, we have to change actually our tool. So our tool has the target recognition, and it's two components, right? And this cutting endonucleases activity, which is actually the cutting of the DNA. But this target recognition is a very important one because it will bring all this protein or the enzyme in the place that we want to actually work in the DNA, in the genome. So, but we can, for example, get rid of the endonuclease activity and add another activity. It's an effector molecule. This molecule can do other things, so we can actually keep the target recognition and do other things in the DNA. So an example of this, for example, we have the normal gene editing or base editing. The gene editing is what I just told you before. We have a guiding sequence and two cutting activities. The base editing, we don't cut anymore the DNA, but together with the Cas, there is another effector which can change one base or one nucleotide to another. For example, we can change a thymine for cytosine, a guanine for an adenine. So some of these, it's a more precise modification of the gene, and it's just punctual. Another, for example, we can modify also directly the RNA using another Cas, which is a Cas13. There are different Cas, just not the 9. The Cas12 is also used for gene editing, for example. And another application is gene regulation. You can, for example, use like a Cas9 which doesn't cut. It's called dead Cas9. And the effector is actually one protein which can activate or deactivate the promoter directly. So we don't get rid of the codifying sequence or mutating it, but we can actually turn off and on this sequence. Another one is, for example, with epigenome editing. So this modifier, the effector, is actually adding or taking off this methyl group. We can actually regulate also the transcription of a gene. And there have been some application of this also in plants. For example, in mice, there is one gene, which is the ILS gene, which has been a base editing. So we can change one nucleotide, like for example is highlighted here, and we can generate an herbicide resistant in the plant. This has been done in mice or in mice, has been done in tomatoes, also in potatoes, changing this single one base, and so we can generate the resistance to an herbicide. Another application, for example, is a gene activation, has been done in Nicotiana bentamiana, which is closely related to the tobacco plant. And this, there is another transcription factor. This transcription factor is an effector which can interact with the promoter. It's a protein which interacts with the promoter, which can activate a gene. So, for example, during the development of the plant, there is a gene participating in the polyphenol biosynthesis, which is actually off, and we want to turn it on to have like the compound in the plant. So this is something that has been done. And we can see here that the expression is higher. And another example is in cotton, when they generate fungal resistance in the cotton, like actually editing one base pair two, or editing actually with knockout this gene. So if you see here in the pictures, the plants that are actually knockout are more resistant to the fungi than the wild type. So with all this, I want to thank you for your attention, and I want to say that even though CRISPR can do so many things, and this was actually just like an overview, we have to take this powerful tool with we have to manage it with care, because ethics is a very important characteristic in scientists. I don't know if there is some questions.
Yes, are there examples of unethical use of the CRISPR technology?
Yes, there have been some examples of unethical use. I haven't talked about animals or human application of the CRISPR-Cas, which actually they have been developed more application in humans. And one example that I can think now, that probably you have read in the news, there was the use of CRISPR-Cas to actually edit babies in China. There was a scientist which was actually using the CRISPR-Cas to decide which features, actually phenotypical features of the babies the couples want to have. For example, blue eyes, blonde hair, I don't know, tall. This is characteristic that we know the genes where they are responsible with, so you can modify this gene in the probably was modified in the embryo, in the early stage of the embryo, or can be also modified in the sperm and the egg. And we can decide which kind of babies we can have. And this was a very big news, like 2 years ago, I think, 2-3 years ago. And later, after a year or two, the Chinese government discovered that these scientists didn't have the permission or the ethical permission to do this kind of experiment. So it was actually fired and banned from the scientific world. So we don't know what he is right now. I think there are other actually applications that can be more useful. I don't know. Ethically, if you want to decide which kind of baby you want to have, I think there are other tools so you can actually discover if there is some genes they are mutated in your sperm or egg. For example, they generate other disease like Parkinson's disease, Alzheimer's disease, like diabetes. This kind of application, I think they could be more useful and probably ethically more correct to actually manipulate than others.
Thanks. And there's another question.
Okay.
In which organism does CRISPR-Cas system naturally occur?
Naturally occur in bacteria. It's a bacterial immune system, and this example is Staphylococcus pyogenes. It's the Cas9 that we all use. The most have been actually used since they discovered. That was the one used in the 2012 from the paper that was published for these two scientists. But there is Staphylococcus aureus Cas9. There is bacteria. There are several ones. Each Cas9 has a different PAM sequence. So sometimes you can use different Cas9 depending on the sequence that you want to work with. That's something that I haven't talked about, but it's a big field, and it's been also engineered Cas9 to change the recognition site of the PAM, which is also the limiting step. But it occurs naturally in bacteria.
Yeah, that's it.
That's it? Okay. Thank you very much.